Hello, I am Dr. Aizaz from Medicovisual.com, and in today's visual lecture we will discuss what is mesenchyme, what is the concept of mesenchyme, and related with this we will discuss what is the source of mesenchyme, what is the function of mesenchyme, and then we will also discuss a common question that I often hear in the comments section, that is the term mesenchyme same as mesoderm? Is Mesenchyme another fancy name of Mesoderm? And along with that, of course, we will also compare and contrast the mesenchyme with epithelium. So let's start the discussion from very basic concept that mesenchyme actually consists of irregularly shaped cells. So if you look here, this is, let's say, one mesenchymal cell. It has a very irregular shape. You cannot say it's a spherical, or it's a cuboidal, or you can say it's a columnar. You cannot define a particular shape. It has very random shape. It can be having any shape, right? So it does not have a proper shape. It is irregularly shaped. Second thing is that these irregularly shaped cells, by the way, these are called mesenchymal cells. So these irregularly shaped cells, they are interspersed, they are loosely interspersed in a gel-like or jelly-like ground substance. So you see, this is the ground substance. So these mesenchymal cells, they are spread, they are loosely scattered here and there within this gelatinous or gel-like, this pinkish purplish one. It is the ground substance, right? So these are the mesenchymal cells. These are loosely spread within a gelatinous ground substance. This is the structure of mesenchyme. Now something important about these mesenchymal cells, that mesenchymal cells undifferentiated cells. These are not properly differentiated. So because they are undifferentiated cells, these are basically also called stem cells, mesenchymal stem cells. They can differentiate and they can form different variety of cells, right? And along with that, of course, the mesenchyme, it is said to be, mesenchyme is called embryonic connective tissue. It's an embryonic connective tissue. The if you know a little bit about the adult connective tissue, you know that a connective tissue has very similar structure. As you see here, we have some cells, and these cells, they are scattered, they are interspersed in a gelatinous ground substance. But the difference of this embryonic connective tissue with the adult connective tissue is that in adult connective tissue, we do not have significant population of undifferentiated cells. Actually, there are some undifferentiated cells in the adult connective tissue as well, but most of the cells in the adult connective tissue, they are well differentiated. They do form fibroblast or chondroblast depending upon the type of connective tissue. They are, most of them, not all, most of them are differentiated cells. Most of them are well differentiated cell. But in this embryonic connective tissue called mesenchyme, it is rich in undifferentiated mesenchymal stem cells. And the third property is that they have irregularly shaped, they are loosely arranged, and because of that, because they are loosely arranged, they can easily move. So mesenchymal cells can easily migrate. They have excellent migration potential. They can easily migrate. So wherever migratory cells are required in the embryo, these cells come to the help, right? So mesenchymal cells, they can easily migrate. Now compare this with something which is stark opposite, which is totally opposite to these mesenchymal cells or mesenchyme. That is called epithelium. So if you come to the epithelium, here you see that when we talked about mesenchyme, we said that it has irregularly shaped cells. These are irregularly shaped cells. But here, in case of epithelium, we have properly shaped cell. For example, here there is this proper border. You see, these are properly shaped cells. So these are properly shaped cells. In case of epithelium, epithelium consists of properly shaped properly shaped cells. Here we said that these are loosely arranged, loosely interspersed in a ground substance, but here they are tightly packed together. They are tightly adherent with neighbors, tightly adherent with their neighbors. Even they have some certain proteinaceous connections with their neighbors. These are tight junctions, zona adherence, and other those details we will talk in histology. So these are not loosely arranged, these are properly shaped. You see here we have cuboidal cells, right? So these are properly shaped. Secondly, they are not loosely arranged, these are tightly adherent. Properly shaped compare with irregular, tightly adherent compare with loosely arranged mesenchymal cells. So, and of course, because they are tightly packed together, they are tightly bound with the neighbor, you know, it will be very difficult to, let's say, pull this cell from here, pull this out from here, and to make it move from here, to migrate it from here. They are sort of fixed here, they are adherent here. So it means that they are difficult to migrate. So these are exactly opposite type of tissues. Epithelium is exactly opposite to mesenchyme. I hope you got the concept up till now. So now let's move forward and let me tell you something interesting. So here what do you see? Here you see this ground, and on the ground you see these pebbles or stones. These stones are scattered here and there loosely and randomly on the ground. This is just like mesenchyme, you see. And here you see that this is a brick wall, and this brick wall consists of these bricks, and bricks are tightly adherent with each other, forming this barrier or wall. So this is like epithelium. This is like epithelium. This brick wall is like epithelium, but this pebble arrangement on the ground is like mesenchyme. Now if I ask you, uh, what will be easier? Will it be easier to take a brick from here, pick a brick from here and move somewhere else, or will it be easier to pick a stone or pebble from here and move it somewhere else? So your answer will be, of course, these pebbles. So I hope now you understand that why these are easier to migrate. They have excellent and remarkable migration potential, and these are fixed mostly. They are difficult to migrate. Of course, we will see later on that they can interconvert. These pebbles, they can interconvert into this brick wall, and this brick wall can interconvert into these pebbles. So that is the concept of transition, epithelium to mesenchymal transition, or mesenchymal to epithelium transition, that we will see later. So now let's see what is the source of mesenchyme. So here is the trilaminar germ disc, and let's say neurulation is going on here. So let me label these things for you. So here we have the ectoderm. This is the ectoderm. Then we have this inner one, or to be precise, this one is the ventral one. This is the endoderm. And here you see this endoderm is sort of folding to form the neural tube, and with neural tube at the crest, or at this margin of neural tube, some cells will migrate from here, some cells will begin to migrate here, and these migratory cells are basically neural here as well. These are neural crest cells, and basically neural crest cells, ultimately they are arising from ectoderm, so they are ectodermal in origin. And of course between the ectoderm and endoderm there will be mesoderm. Mesoderm will be forming these layers, these compartments, the paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. So I hope you got the concept. Bilaterally this will be the situation here, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. So this is the situation right now. So now, what is the source of mesenchyme? What is the source of mesenchyme? The single most important source of mesenchyme that you must remember is mesoderm. So the most common source of mesenchyme is mesoderm. Most common source of mesenchyme is mesoderm. Please remember this. But mesoderm and mesenchyme, they are not the same. Mesenchyme is the migratory cells that develop mainly from mesoderm. These are the migratory cells, mesenchymal cells. These are the migratory cells that develop mainly from mesoderm, right? But mesoderm is the middle one. Meso means middle, and it is basically the middle layer of trilaminar germ disc. So we have 1, 2, and 3 trilaminar, tri-layered. So we have tri-layered or trilaminar germ disc, and middle layer of this trilaminar germ disc is mesoderm. So middle layer is most common source of mesenchymal cells. Since this is the most common source of mesenchymal cells, even some very famous textbooks, they erroneously do this practice, which is not good actually. What they do is that they use the term mesoderm interchangeably with mesenchyme. This is totally wrong. These are not same thing. Actually, not all the mesoderm is in the form of mesenchyme, and not all mesenchyme comes from mesoderm. Later I will tell you, just now I will rather tell you that what is the other sources of mesenchyme. So please make this concept very clear in your mind. Mesoderm is not mesenchyme. But because major source of mesenchyme is mesoderm, that is why many textbooks, many authors, they erroneously interchangeably use these two terms, but it is not appropriate to do so. Please get this point straight in your mind. Now, besides the mesoderm, the second source of mesenchyme is neural crest cells. Neural crest cells, and of course, as I've told you, neural crest cells, they are coming from ectoderm. So we can say ectoderm via the neural crest cells is the second most common source of mesenchyme, right? And finally, transiently and rarely, there is very little mesenchyme that also comes from endoderm. So there is very minor contribution towards mesenchyme is by the endoderm, and it is usually transient, so I will not discuss its detail. So mainly the mesoderm is the major source, number one, and second one is the ectoderm via the neural crest cells. Now here again I will stress these 2 points. I will write it here that please don't confuse the mesenchyme with mesoderm. They should not be ideally used interchangeably, because not all mesenchyme comes from mesoderm. As I've told you, the other sources is neural crest cells, and very little may also come from endoderm, right? So although this is mesoderm is major source, but there are other sources, so it is inappropriate to interchangeably use these two terms. Secondly, the second reason that these terms should not be used interchangeably is that not all mesoderm is in the form of mesenchyme. The major one is paraxial mesoderm, which forms the sclerotome and dermomyotome, which actually contributes a lot towards mesenchyme. But then we have this classical lateral plate mesoderm. So if we draw a paraxial mesoderm, then intermediate mesoderm, then we have this lateral plate mesoderm, which has this type of arrangement, this squamous type of arrangement of cells. These are more of epithelial in nature. These are more of epithelial in nature, right? So actually, these are not in the form of these cells of lateral plate mesoderm. They are not in the form of mesenchyme, but they actually do contribute towards the mesenchyme. Although they are not in the form of mesenchyme, they actually do contribute towards the mesenchyme. Well, that might sound confusing for some of you, so let me tell you. I have already told you about this. I had given a sneak peek that epithelium and mesenchyme might undergo interconversion, that is called transition. So for example, if this epithelial cell has to form the mesenchymal cells, what it will do is that first of all it will break its connections with its neighbors. So let's say here we have a cell and here we have another cell. So between these two cells they are sort of shaking hands, right? They are having these epithelial interconnections, right? So first of all, there will be down regulation of these proteins that are attaching them with their neighbors. So what will happen, that these cells, they will detach from their neighbors. And secondly, there are certain proteins within the cell that causes a particular shape of these cells. So these cells will lose the shape. And thirdly, they will start secreting a ground substance to form the mesenchyme. So epithelium can be interconverted into mesenchyme. This process is called epithelium to mesenchyme transition or epithelium-mesenchymal transition, EMT, epithelial-mesenchymal transition. Likewise, opposite can be done. These certain proteins can be produced which make particular shape of the cell, and then certain junctions or certain tight zona adherence and the other molecules can be expressed on the surface of these cells, which cause the neighboring cell to meet up with each other when they form the cellular processes to meet with their neighbors, right? So they can be converted back to mesenchyme, can be converted back to epithelium. So mesenchymal epithelial transition may occur. So epithelium to mesenchyme is EMT, and from mesenchyme back to epithelium is MET. So with this process, these cells, these lateral plate cells, they proliferate, and actually some of them, they undergo epithelium to mesenchymal transition to form mesenchyme. And this mesenchyme, it goes to different places. It goes to the limb buds, for example, to contribute towards the formation of skeleton. It also contribute towards the formation of the connective tissue of the muscles of your limbs, and so on. There are many contributions. You will learn those in musculoskeletal embryology. But the thing is that lateral plate mesoderm, although itself it is not in the form of mesenchyme, as I told you, itself it is not in the form of mesenchyme, but it not only can, it actually contributes towards the formation of mesenchyme. So please understand these things. So in a nutshell, basically, wherever the migratory cells are required, cells have to migrate. For example, okay, let's come up with a simple example. Let's say, so for example, okay, here we draw the body, simple body, and here for example we have limb bud, and then we have what's here, paraxial mesoderm, and it is forming the somoid. Let's draw a single side only, an intermediate mesoderm and lateral plate mesoderm, right? Now you know that we have sclerotome and dermomyotome, right? So from the myotome, actually we have this. So we have these hypomeric muscle cells here. These are the epimeric here, which are going to form the muscles of the back. And then we have hypomeric muscle forming cells, hypomeric muscle forming cells. So these hypomeric muscle forming cells, they are in the form of mesenchyme. So they will migrate from here and they will come near this lateral plate mesoderm, and from here they might go, some of them they actually go into the limb bud to form the muscles of the limb. So you see they have this migratory potential. So that is why it's an excellent tool. Mesenchyme is an excellent tool for the body. Wherever the migration is required for the production or for the development of some organ or some tissue, wherever the migration is required, our body uses this tool, this mesenchyme, for the development of such organs. Actually, to be very precise, most of non-epithelial structures develop from what? From mesenchyme. Most of the non-epithelial structures develop from mesenchyme. For example, you know, bone is a very specialized kind of connective tissue. If you don't know, you will understand it in histology. This is a very basic kind of lecture, and I am deliberately skipping the detail. Then cartilage is another interesting type of connective tissue like substance. It is a specialized connective tissue, right? And then we have dermis, which is the connective tissue of your skin, right? And then of course muscles. These are technically not connective tissue, but these are of course non-epithelial structures. And of course our adult connective tissue, that also arise from mesenchyme. So most of the non-epithelial structures, they develop from the mesenchyme. Even some epithelial cells also develop from mesenchyme. So it is very important structure. Some epithelial cells, they also develop from mesenchyme. A classical example, here I will tell you a single example. A classical example is renal tubules. If you go and watch my lecture, it is free on YouTube. Watch my lecture on the development of kidneys. You will learn the concept of metanephric blastema. And then, you know, metanephric blastema, they put a cap on developing renal tubules, right? And these caps, they form the renal tubules and different structures related with kidneys, and these tubules are basically epithelial in nature. So even some epithelial structures, they also develop from the mesenchyme. So that is why mesenchyme is very, very important structure. So in a nutshell, in a single sentence, if someone asks you that what is mesenchyme, so you can tell that mesenchyme consists of undifferentiated, irregularly shaped cells that are loosely interspersed in a gelatinous ground substance. That is a single sentence definition of the mesenchyme. And why it is important? It is important for development of many important organs in our body. Most of the non-epithelial structures of the body, they develop with the help of mesenchyme. And thirdly, what is the most important source of mesenchyme? Your answer should be that single most important source of mesenchyme is mesoderm, and second most common source of mesenchyme is neural crest cells, and there is almost little to no contribution from the endoderm, right? So what it means is that all three germ layers, they contribute towards the mesenchyme, but mesoderm and ectoderm are the most important sources. Also, please do not use the mesenchyme and mesoderm terms interchangeably. Well, I am saying this, but I think I might have used this term interchangeably somewhere. But this is wrong. This is wrong way to do so. Even if I did it, I don't know if I did it, but even if I did it, I can say confidently I did it wrong. So I have no problem in confessing that. Anyhow, so not all mesenchyme comes from the mesoderm. Some comes from neural crest cell and some from endoderm as well. That is very rare. And secondly, not all mesoderm is in the form of mesenchyme. For example, lateral plate cell, it is in the form of epithelium, but despite being in the form of epithelium, it does contribute towards the formation of mesenchyme through the process of epithelium to mesenchymal transition. And finally, before closing this lecture, let me ask you a question. You know, in the during the start of third week third week of development of the embryo, the epithelium to mesenchymal transition process occurs very widely. And you know when it occurs, that you will tell me in the comment section below, that during what process epithelium to mesenchymal transition process occurred at the start of third week of development. That is your assignment. You have to answer this question in the comment section below. Thank you so much for watching this video. That was all for today. If you have a question, feel free to ask me in the comments below.